In last video, we have done the variant call from haplotype caller and did some hard filtering in the resulting variants. And the next step is to do the annotation and effect prediction. Just to let you get familiar with the term we're gonna see in the annotation result file, I want to first go through the types of genetic variations in human genome one more time. So there are many types of genetic variation. The most serious one is chromosomal aneuploidy. It's the abnormal number of chromosomes in a cell. For example, a human cell having 45 or 47 chromosomes instead of the usual 46. One example is Down syndrome. These children have 3 copies of chromosome 21. There are some words that are easy to mix up. One is mutation, the other is polymorphism. Let's say in a population, for example, all the people in the world, if this variation is less than 5%, we call it mutation. If it's more than 5%, we call it polymorphism. And sometimes we use a cutoff of 1%. This is the definition, the difference between these two. And the variation and the variance usually mean the collection of both mutation and the polymorphism. Structural variation and copy number variation. This include a large section of deletion, a large section of insertion, both novel sequence insertion or mobile element insertion, inversion, or duplication, depends on whether it's the duplicated part is relatively far away, it's interspersed duplication, or is next to each other, then it's tandem duplication or tandem repeats. There are 3 different approaches using next generation sequencing reads to detect structural variation. One is using the read pair or pair-end reads. Depending on the insert size and relative to the expected insert size, then we know whether it's a deletion or insertion. Also, depends on the read depth on a large region. This method is usually used to detect copy number variation, whether it's a deletion or amplification. And if we look at a particular read, if it can detect a certain junction or split point or the break point, then we will know the break point of the deletion or insertion. The short insertion and deletion is called indel. This may occur in the intergenetic region or intronic region or coding region. If it's in coding region, it may cause 2 different consequences. One is cause frame shifting, the other one is not cause frame shifting. Frame shift is, for example, the deletion of this C nucleotide will shift the reading frame of the codon, translate to protein by 1, and it will generate a totally different peptide. So at this time, not only this sequence of protein will change, but it will often introduce a premature stop codon, so the protein will not be translated. Single nucleotide variation is very common in our human genome. Generally speaking, each of us has 3 million single nucleotide variation in the genome. That is equivalent to a frequency of 1/1000. It may appear in intergenetic region or intronic region or promoter region or coding region. It may also occur in the UTR regions or non-coding RNA regions. If it's in the coding region, it has several possible consequences. One of the most serious is that it will introduce a stop codon, so the protein will terminate early, or the protein will not be translated, or the protein will produce a truncated version. This is usually called stop gain or nonsense mutation. Sometimes it will also cause a deletion of the stop codon. That is, the final protein will be longer than the original one. But it may also be the case that it cannot be translated. The most commonly known mutation that affects amino acid is called a missense mutation or a non-synonymous mutation. These non-synonymous mutations are currently the most studied one. The main reason is that it changes the amino acid, changes the sequence of the protein, even though they add up to be only less than 2% of the human genome. But they are involved in about 50% of mutations that we know in human genetic disease. There are also some mutations that occur in the exon region. But because this codon is degenerative, so some of them are synonymous. That is, it does not cause amino acid change. This is called synonymous or silent mutation. Finally, in the junction site where the intron and the exon meet, some mutations can affect splicing, and they may also have serious effects on proteins. The last part of our computer lab is the functional annotation on the resulting variant file. So we'll use SNP-if to add functional annotation to a set of variants. So I will not do the operation in this video. Rather, I will leave this as a homework. You can just follow the command I listed here. You can also look at the documentation file and some website about the SNP-if. And for the homework, I will be looking for the result file of this mother HC, annotate VCF file in the OSC ovens.